Dear students, this is our second lecture on medicinal chemistry, and in today's lecture we will discuss about physicochemical properties of various targeted molecules which can act as drugs. Uh, Medicinal chemistry basically is an interdisciplinary science, and it can be thought of as at an interaction with the chemistry and pharmacology. And here, in case of medicinal chemistry, we need to undertake the identification, synthesis, and development of new chemical compounds which are suitable for medical or pharmaceutical use. And for the complete medicinal chemistry to be understood, we need to understand the branches of these types of chemistry and pharmacy, which in combination will make us a better medicinal chemist. And most important among these branches, they are organic chemistry, phytochemistry. Phytochemistry means natural product chemistry, biochemistry, physical chemistry, statistics, computational chemistry. Computational chemistry these days is very important because this tells us about the QSIR studies for different types of drug molecules. Then molecular biology, toxicology, and pharmacology, which will tell us about that how much active our targeted drug molecule is. Then, as many of the drug molecules these days are being prepared in the laboratory, so toxicology is also an important branch which will tell us about the toxicity of the given compound which is thought to be a drug molecule. Now, as far as biological activity of the targeted drug molecule is concerned, this will mainly depend on its physicochemical characteristics. And some of the common physicochemical characteristics, they are hydrogen bonding, chelation, oxidation reduction potential, dissociation constant, bioisothermism, surface activity, and steric features of the drugs. Now, there are so many physicochemical parameters, and it is difficult to correlate that which biological property is related to which physicochemical processes. So, therefore, in case of different drug molecules, we need to identify the different atoms, functional groups, or even the entire molecule that which will determine the physical and chemical parameters which are involved in the drug molecule. So basically the biological activity may be dependent on any one of the physicochemical parameter, or it may be a combination of more than one physicochemical parameter which will determine the biological activity of that targeted drug molecule. So first property is hydrogen bonding, which is very important for a molecule to act as a drug molecule. Because as far as drug molecules are concerned, there is a minimum aqueous solubility which is required for a molecule to act as a drug, because that drug has to be transported from the site of action to the receptor molecule. Therefore, hydrogen bonding, which will affect the solubility of the drug molecule, therefore is an important aspect for the biological activity of that compound. And as you know, that hydrogen bonding basically is a bond between the hydrogen atom, which is an electrophilic atom, and it will form a bond with the an other atom, which is usually electronegative atom. So most commonly occurring hydrogen bond is between hydrogen and the electronegative atoms like fluorine, oxygen, and nitrogen. Whereas to some extent the hydrogen bonding can also be shown by sulfur and chlorine atom. And this hydrogen bonding is of two types: intermolecular hydrogen bonding and intramolecular hydrogen bonding. Intermolecular hydrogen bonding occur between two or more molecules. For example, in this case, if we are having different water molecules, now one water molecule, oxygen of one water molecule will form a bond, hydrogen bond, with the hydrogen of the other water molecule. So like this type of bonding, which is between different molecules, is called as intermolecular hydrogen bonding. Whereas intramolecular hydrogen bonding is where hydrogen and the electronegative atom involved in hydrogen bonding, they are present within the same molecule. So for example, in case of salicylic acid, hydrogen of this OH group and oxygen of this carboxylic group, they are involved in the hydrogen bonding, and both the atoms, they are present in the same molecule. So this is an example of intramolecular hydrogen bonding. Similarly, orthonitrophenol, hydrogen of this hydroxyl group and oxygen of this coordinate bond with nitrogen. So both of these, they are present in the same molecule. So this is again an example of intramolecular hydrogen bonding. Then the strength of the hydrogen bonding will depend upon the type of electronegative atom which forms a bond with the hydrogen. More the electronegativity, more will be the bond strength. For example, fluorine is the most electronegative atom. This is having the highest bond strength, and then this will also depend on the structure of the molecule also. So as the electronegativity decreases, the bond strength also decreases. And as I have told you that there is a minimal aqueous solubility required for all drug molecules to be transported from the site of action on a receptor. Therefore, strength of this hydrogen bonding will depend also on the type of solvents which are involved in the reaction. So the hydrogen bond strength of O, oxygen and hydrogen for acetic acid dimer in vapor state is 7.64 kilocalories per mole. But if we use benzene as the solvent, then it is 4.85 kilocalorie per mole. So that means hydrogen bonding is dependent of the physical state of the molecule also. Similarly, you can see that in water the hydrogen bond strength is 4.5 kilocalorie per mole, whereas in ice it is 6 kilocalorie per mole. And in water this is the liquid physical state, whereas ice is the solid physical state of the molecules. So a lot of factors, they will determine that what will be the strength of the hydrogen bonding. And the drug receptor interaction, which will be determined on the basis of hydrogen bonding, This is closely related to the importance of these bonds in determining the physicochemical properties of different drug molecules which are being considered for the biological activity. Then the physical state of substances such as water, DNA, protein, and various drug molecules, they are maintained by hydrogen bonding. So, for example, you can see that in case of complementary base pairs like adenine, thymine, also there is an involvement of hydrogen bonding. So, because this is taking place in two different molecules, so this is an example of intermolecular hydrogen bonding. Guanine and cytosine, they are also complementary base pairs, so there is a hydrogen bonding between these intermolecular hydrogen. Then physical and chemical properties, as I've told you, of a compound, they may change because of the hydrogen bonding, and this can be shown from few examples also. And this extent of hydrogen bonding will also affect the biological properties of different types of compounds. For example, if we compare the biological activity of this compound antipyrine with this compound 1-phenyl 3-methyl 5-pyrazolone, and this is if you see the structure of these two molecules, the structure is similar. Only difference is that here this nitrogen is having hydrogen atom attached to it, whereas here that hydrogen has been replaced by a bigger group, that is methyl group. So basically this is 3-methyl, where this is 2,3-dimethyl compound. So if we see the structure is similar, but if we see this molecule, because of presence of this hydrogen, this can form a hydrogen bond with another molecule of this type, and this hydrogen will form a hydrogen bond with the carbonyl of the other molecule. So likewise, because of this intermolecular hydrogen bonding, a linear polymer of 1-phenyl 3-methyl 5-pyrrolazone will be formed. And because this is now a polymer, because of high molecular weight of this linear polymer, it cannot pass through the biomembranes. So that means this molecule cannot be transported from the site of action to the receptor. Therefore, it has no analgesic activity, right? Whereas when this hydrogen is replaced by methyl group, then no such linear polymer is formed. And because of that, this molecule does not show any hydrogen bond. And in the absence of hydrogen bonding, this is soluble in water and moderately soluble in ether. Whereas because of the hydrogen bonding, this is insoluble in water and slightly soluble in ether. And this shows a good analgesic property, whereas this does not show any analgesic property. And this shows good analgesic property because this can be transported easily from the site of action to the receptor molecule. So this example clearly shows the effect of hydrogen bonding on the biological activity of the molecules. Similarly, if we compare the biological activity of orthohydroxybenzoic acid and para-hydroxybenzoic acid, you can see that this molecule is having an intramolecular hydrogen bonding, whereas this molecule having hydroxyl and carboxylic acid group attached at 1 and 4 positions, they have only intramolecular hydrogen bonding which is involved. You can see over here, this is an example of intramolecular hydrogen bonding, whereas this is a dimer of 1,4 benzoic acid, para-hydroxybenzoic acid. This shows an intramolecular hydrogen bonding, and because of the intermolecular hydrogen bonding, it shows more solubility in water, whereas because of intramolecular hydrogen bonding, this is less soluble in water, and because of that, this is having a good antibacterial activity, whereas this is having a low antibacterial activity. So again, the extent of hydrogen bonding affects the biological activity of different types of molecules. Then next property is chelation. Chelation, or this is called as formation of coordination complexes. And chelation results from donor receptor mechanism, that is one molecule will donate electrons, whereas other molecule will accept electrons, and this is again similar to Lewis acid based reaction. In Lewis acid based reaction also, there is one molecule will be donating electrons, donating protons, whereas other molecule will be accepting protons. So therefore the compounds that are obtained by donating electrons to metal ions with the formation of ring structure, they are called as chelates, and the process is known as chelation. The most common examples, they are the biological molecules or medicinal agents which can develop chelating structures by forming a ring structure with the metal through coordinate bonds. For example, this is amino acid glycine anion. You can see this is oxygen can donate electron, then nitrogen is also having a lone pair of electrons which can be donated, and with different metals, say copper 2 plus ion, one electron with this one, they will form a bond, a ring structure, or glycine copper complex will be formed. So this is called as a chelating complex of glycine with copper, and the stability of this complex will be determined by the stability constant. More the value of this stability constant, higher will be the stability of the complex. Then most commonly used electron donating groups, they contain oxygen, nitrogen or sulfur atoms, whereas the metal atoms which act as electron acceptors, they are usually bivalent and trivalent metal of the transition group. And stability of different metals in the formation of chelating complexes is Fe3, that is ferric ions. They are the most stable ions which can form chelating complex, followed by mercury, copper 2 plus, aluminium 3, nickel, lead 2 plus, copper 2 plus, zinc 2 plus, ferrous, cadmium 2 plus, manganese 2 plus, magnesium 2 plus, and calcium 2 plus. So the complex formed with ferric will be the most stable, and the complex formed with calcium will be the least stable in terms of stability of these different types of metal ions. Then the commonly used multidentate ligands, they are ethylene diamine. Here you can see both these amino groups. Nitrogen will have lone pair of electrons, they can act as ligands, and then dimethyl glycine, nitrogen oxygen, they can act as coordinating agents. Then bipyridine, these nitrogen oxygen, nitrogen in case of 8-hydroxyquinoline, 110 phenanthrolene. This can have these two nitrogen having lone pair of lactones acting as ligands. Then ethylene diamine tetraestic acid is a very good complexing agent. This can form a complex with bivalent metal ions very easily. Then in case of enzymes and biomacromolecular compounds, there are different types of metal ions which are required for the action or activity of these enzymes or biomacromolecules. For example, in case of vitamin B12, we know cobalt metal ion is present, which will form a chelating complex with the rest of the structure of the vitamin B complex. Similarly, carboxypeptidase, it contains cobalt. Oxidase enzymes, they contain copper. Then porphyrin enzymes, they contain iron. Hemoglobin, this also contains iron. Chlorophyll, magnesium. Chloroplast, manganese. Then xanthine oxidase, it contains molybdenum. Aldehyde oxidase, it contains molybdenum. Urease enzyme, it contains nickel. Then carbonic oxidase, this contains zinc. Alcohol dehydrogenase, it contains zinc. So all these types of enzymes which are present even in our body, they contain these metal ions with whom they chelate to form a strong chelating complex, coordinating complex, which will act as a drug for different types of diseases. Then metal binding hormones like thyroxine, insulin, histamine, epinephrine, and norepinephrine, they can also bind metals very easily to form chelating complexes. And in clinical practice, the chelating agents, they have been primarily used as antidotes in heavy metal poisoning. So metal poisoning can take place in the body due to different types of metal ions. And when these chelating agents, they are given as drugs in the body, they will form a complex with those metal ions, and those metals, they can be removed easily from the body, hence they can be used as antidotes. Then the chelating, any chelating agent with sufficiently high stability constant in vitro could be used as an antidote, but some limitations are there. For example, if we take the case of ethylene diamine tetraestic acid, EDTA, it has a powerful chelating effect, and it can displace toxic heavy metals such as lead and mercury from the cellular layer. But at the same time, the selectivity of EDTA is not very high. That means in addition to forming complex with lead and mercury, it will also form a complex with other essential metals which are present in the body like calcium. So when it tightly binds with calcium metal, that means in addition to acting as an antidote for lead and mercury, it will also remove the essential metal, that is calcium, in the body. So therefore, when EDTA is being used as an antidote to remove these metal ions, we need to give some additional drugs to the patient undergoing therapy for these antidotes in the form of, say, disodium calcium salt. An additional drug is to be given so that there is no excessive loss of calcium from the body during this EDTA therapy for that patient. Then other examples of different diseases where these complexing agents, they can act as antidotes. For example, if in the body there is iron deficiency, that is anemia, this can be used by iron complex of EDTA in place of ferrous sulfate or ferrous gluconate. So we can give iron complex of EDTA from where iron will be taken up by the body. Then pernicious anemia, this is treated by using vitamin B12, and this vitamin B12 we know is a naturally occurring cobalt complex. Then deferoxamine is basically an amide which act as an antidote for iron poisoning. If there is iron poisoning in the body, we can give deferoxamine. This will form a coordinating complex with the iron molecules which are present in the body, and those will be removed from the body. Then dimercaprol, this is used for the treatment of lead poisoning. You can see the structure of dimercaprol, and because of the presence of these hydroxyl and thiol groups, they can easily coordinate with the lead molecule, so this can act as antidote for lead poisoning. Similarly, L-penicillamine, this can be used in treatment of copper and Wilson's disease. So Wilson disease is basically an antidote for copper, which is accumulated in our liver, brain, and other vital organs of the body. So because of the presence of these atoms, that is thioamine and the hydroxyl group of carboxylic acid and carbonyl group, they can act as ligand molecules where copper can be chelated, so that can be removed from the body. Then similarly, many antimicrobial and antineoplastic agents, they are believed to exert their action by means of complex formation with the DNA base pairs. So most commonly used agents, they are ethidium, proflavin, quinacridine, doanorubicin, adriamycin, actinomycin D, then hydralazine. So all these compounds or the drug molecules, you can see they have large planar aromatic structure, right? And because of this planar aromatic structure, they can be inserted between the planar base pair assemblies on the DNA double helix, and this type of interaction between the DNA and these planar aromatic compounds is called as intercalation, and these drugs, they are called as intercalating drugs. So these are the most commonly used intercalating drugs, which will be acting as either antimicrobial drugs or antineoplastic agents. But these intercalating drugs, they have few side effects also. That is, for example, hydralazine, this one, because of the presence of these nitrogen atoms, this can strongly chelate with the iron metal present in the body, and this will lead to the anemia or iron poisoning. Similarly, 8-hydroxyquinoline and its analogs, they generally act as antibacterial and antifungal agents, but they can make a complex with iron or copper metals present. So this is the literature which has been used for preparing this lecture. Thank you very much.